It's been a busy week in the lab with lots of projects underway, but none are quite ready for their own video, so I wanted to go over progress and do some updates as well as answer some of your questions. Last week we looked at building two important pieces of lab hardware, and I'm happy to report that they work perfectly. I just got new media in, so I wanted to test it all out, and I needed to propagate some organisms anyway. I mixed up some LB and malt agar, and then some LB broth for liquid cultures later. All the agar plates were poured, and once cooled, it was streaking time. I didn't film all of it, but here I'm doing a few different things. First, I'm setting up some new D47 wine yeast so I can retry the yeast transformation, and I'm streaking that out on malt agar. The next is another yeast I'll be using for the spider beer project. The species is K. marxianus, and normally this is a spoilage yeast and not typically used for brewing due to what those adventurous enough to try describe as heavily scented and taste like nothing. Eventually I'd like to transition the project to a more readily available yeast, but this is what I have for now. The reason this yeast was chosen is because it's nearly genetically identical to the species I'm supposed to use with the integrating plasmid, so hopefully it's close enough to work. If not, we'll tweak the plasmid to work in normal brewer's yeast instead. I also did a test streak of some yogurt in preparation for the Glowgurt project, then propagated some E. coli that have a bioluminescent plasmid in them. Everything was loaded into the incubator, and within a day, everything had grown. Here's what 2 days did to the K. marxianus culture. I'm so pleased with how well this works. This is really going to speed things up. Another piece of hardware I've been working on showed the first signs of success, but still needs work. Fluorescent microscopes are really amazing tools, but are also extremely expensive, which I think is a shame. So like the polarimetry mod I showed in a previous video, I'm trying to modify my microscope to work as a fluorescent scope. To do this, I'm using a special dichroic mirror, which is a type of filter that only lets certain wavelengths through and reflects the rest. In this case, it reflects blue light, but passes everything else. With this in the optical train, I should be able to shine UV light up through the bottom where the white light normally is, and only emitted light from the sample gets through the filter and all the UV is reflected away. In a professional scope, it looks like this, where the colors are sharp, as is the detail, but in the homebrew version, it's more like this. Everything is kind of yellow because of where the reflection point of this mirror is. In professional scopes, they have more expensive and versatile filters and higher quality UV light sources, so only things properly glowing show up and you can see more colors, whereas here there's a vague haze. That said, when I loaded up some fluorescent yeast, they all seemed to glow when I put the UV light on them. One issue with this is because the emission peak of GFP, which is what makes the yeast glow, is so close to the reflection point, I think that most of the light is actually being lost. When I use the most powerful lens on my microscope, the glow is mostly gone or very dim, so I think that's going to need to be improved. One way around this is to just use a different fluorescent protein. For example, I've got some red fluorescent protein coming in soon, so that will give a much clearer test to see if this actually works, and is able to pass emitted light and reflect UV properly. For now, I'm going to keep tinkering with it and see if I can find a better filter. Someone recently suggested polarizing the UV light, so I might give that a try. But for the $10 this filter cost, it could be great if you just choose different fluorophores. I'll post a longer video once I've sorted these issues and done more testing. Finally, the last and biggest item has been starting the spider beer project. Before we get started, I'd like you to meet my collaborator on the Silk project, Vesta. We've been working together to design the DNA and roadmap of the Silk project, and we'll be working together in the lab to build it, so you'll be seeing lots more of her in upcoming videos. To start things off, while we waited for the rest of our reagents, we needed to test our equipment, and so we did a semi-test run of the first step of the spider beer project. I say semi-test because it's only a test if it doesn't work. If successful, we'd be able to directly use the results for later steps in the process. First up, a Pcr run to see how the Bento Lab's built-in PCR works. When I was sourcing PCR reagents, I ended up ordering this weird pre-mixed stuff that comes in individual tubes. The tubes in the box are way too big to fit into a PCR machine, so we had to harvest the waxy substance inside to perform our reaction. Suffice it to say, I'll be getting different reagents for future runs as soon as possible. We dissolved and diluted our primers and some of the donor template DNA, and added small amounts of each to the reaction mix, then capped it and placed it into the machine. Once it warms up, the wax melts and the aqueous layer sinks to the bottom and the reaction can get going. We programmed a simple pattern into the machine based on the theoretical numbers we calculated when we designed the primers. We're also going to be running a much more complex program on a higher quality machine, but we'll be going through all the specifics of that in the PCR video. When the reaction is done, we can then test it to see if it worked, and to do that we're going to need to run a gel. Gel electrophoresis is one of those things you end up doing a lot if you get into biology. It's a really useful analytical tool, but also a very simple one. Again, we'll be doing an in-depth video on this process, but the basic steps are as follows. First, we make the gel by dissolving agarose, which is a type of long-chain sugar, in TAE buffer, which can be bought online or mixed by sourcing individual ingredients. I just bought a 50x bottle of the stuff, which should last for ages. I mixed up a 1% agarose and TAE solution, which is then microwaved for short bursts until the solution is clear and everything is dissolved. Don't forget to add gel stain to the solution before microwaving or you won't see anything on the gel later. The gel is then cast. Here we're using the gel box that comes with the bento lab, and Vesta brought her Mini 1 gel box, which was also loaded up. This way we could compare both systems and also test both of them, as this was our first time using both devices. With the gel cast and cooling, we could make our test solutions. We're making them on a small piece of parafilm, since we only need a tiny amount for this. Rather than going through all of them in detail, I'll just say that for our first run, the order was 100 base pair ladder, PCR sample, 1000 base pair ladder, and then the GFP yeast plasmid. For our second run, we switched the order of the second ladder and plasmid. Each solution is mixed, and then the gels are prepared to receive them. In the case of the bento lab, the rubber stoppers are removed, as is the comb. For the mini one, the combs are removed and the tray is placed into the electrode box. Both boxes are filled with TAE until the reservoirs on either side are full and the gel is slightly covered by liquid. Then our test solutions are added carefully to each well. After that, all that's left is to turn them on and let them go. It became increasingly clear that the bento lab is much slower, and by the time we'd packed up for the night, it was still only a quarter of the way through. We let it run for another hour and left it overnight, but by the time I came in the next day to check on it, it had turned to goop. So that's going to need an upgrade. The mini one, on the other hand, everything moved through the gel quickly enough that we had time to run 2 gels. So what were the results? Did our reaction work? Well, no, and we learned a few things. First is that something went wrong with our PCR reaction. That's why we're also testing on the fancy machine. If it works, then there's an issue with the bento lab and it may need to be recalibrated. If not, then something's wrong with either our primers or reagents and we'll need to redesign and reorder them. Next is that we need to adjust the gel concentration to a higher percentage to slow down the DNA in the mini one. This will give better separation of the bands so we can see things better and impede the DNA a little bit so it won't shoot through the gel as quickly. Also, we need to tweak the bento lab to put out more power as everything moved way too slowly, and if that's not possible, I have a secondary power supply we can use. I also need to tweak the gel box itself as it tended to be a bit leaky and the lid fogged up so you couldn't see what was going on. I may make a new acrylic box, or if I'm lazy, I'll borrow a trick from a friend and just use Tupperware containers. I really like the mini one lid style, so I may adapt that as it's super easy to see through. So while it's a bit disappointing that it didn't work on the first try, it's also not surprising. We'll keep at it and hopefully have more updates soon. If you want to follow along, then be sure to head over to my other social media platforms, especially Instagram, where I'm posting regular photos. Moving on, some quick updates. For the approximately 8 quintillion of you that asked, the Styrofoam worm project is officially restarted. We'll be following the original paper where they isolated the bacteria from the worms and grew it separately for Styrofoam breakdown. First step is to feed the worms some Styrofoam for 2 weeks so their gut flora adjust accordingly and the Styrofoam capable ones get boosted. So stay tuned for more on that over the next few months as it's a rather slow process. Next, just a quick heads up that I just overhauled my website and I think it looks much better now and hopefully provides some better clarity to the sorts of things we do. I'll be continuing to update it as more of the big projects get completed and started. And finally, before we get to the questions, I just want to give a big shout out to my amazing patrons and now channel members, as your support is what allows us to keep all this science going. It's given us the ability to take the time needed to get this stuff working and produce lots of great content. So thanks. And if you'd like to support the show, then either click the join button below or head over to patreon.com/thethoughtemporium and consider joining the turnip garden. Okay, question time. Ian asks, do I consider the Thought Emporium my job or my hobby? Well, thanks to the support of my patrons, making videos has become my full-time job. They say that if you do what you love, you'll never work a day in your life. The reality is that if you do what you love, you end up putting in more hours than you would at, say, a standard 9 to 5. I spend an absolutely enormous amount of time on videos and research and have had to teach myself to take a day off once a week so I don't burn out. But I love doing it and I look forward to many more videos. And best of all, it funds the research I've dreamed of doing for years. All I have to do is talk about it or show it on camera. It's also given me an amazing opportunity to not only talk about and show how to do science, but also show when things go wrong or don't work, something that doesn't really show up in scientific publications. Moving on, Andrew asks about my creative process and how I go about coming up with things for videos or topics I want to cover in my research, and the research techniques I use to find info and get around dead ends. This is a big question, so this will be the last one. The short answer is, I read. A lot. Throughout the day, I'll have conversations that may spark a question, or I'll see something go by on my newsfeed that seems interesting, and then it's down the rabbit hole to find the answer. First up, read Wikipedia to get a gist of what's going on and to start figuring out what the keywords of the topic are so I know what to look up in deeper searches. Then maybe check the references on Wiki and read any articles that seem helpful. After that, I'll start looking for papers on the subject, usually on Google or Google Scholar. I'll skim the abstract, and if it seems like it might answer my question, I'll find ways to get the whole paper if it's behind a paywall, or just read it if it's open access, as all papers should be. Then the cycle just repeats until I either have enough information to proceed or I hit a wall. Sometimes through all of this, I'll find something really cool that I think deserves a journal club video. Others, it's an effect I want to recreate because it seems so absurd that I need to see it for myself. Others, it's something where there just isn't any info, and the only way to find out is if I actually try the thing myself. The only times this is different is if I'm chasing a specific project, like the spider beer, for example. With that, I knew exactly what I wanted to do going in, so I just started digging into every detail about the project I could find. Otherwise, if I'm trying to build a tool like a spectrophotometer, I'll learn everything I can about how it works, what's required to make it, and then start sourcing parts. If the parts list isn't crazy expensive and the tool is worth the time building, then I'll just order things. Once it's arrived, I'll just tinker with it until it works, even if that means putting it down for a bit to think about ways around roadblocks. As to dealing with dead ends, I'm mostly just stubborn. Failure sucks, and if there's some inkling that a thing could work, I'll usually stick with it for a while till I've pushed all the buttons and it's either working or properly dead. But knowing that point is really just a matter of experience. I tend to assume things are still possible for probably longer than I should, but a lot of the times if you just stick with it, sometimes you can get it to work. But eventually you just need to put it down, do something else, and maybe pick it back up later if you've had more time to think. And that's where I think I'm going to leave it for this episode. There's a busy week ahead, so be sure to subscribe and ring that bell to see when I post new videos. Again, a big thank you to my patrons and channel members who keep these science videos coming. That's all for now, and I'll see you next week.